Hey guys, welcome to my first attempt to try and record a video lecture for you. So today we're going to be looking at the first video in a series of genetic mapping, just mapping principles. So in this video today, what we're going to be looking at is effectively reviewing the tools that we'll need for making maps. So these will be the things that we've been learning about all semester. Start using these tools to recognize linkage and then apply these tools to Mendelian principles. And along the way, you may see some screens where it will say, pause that video. So this is your chance to go ahead and, as it says, pause the video and work out some problems that you'll be prompted on. So let's get started. So what is genetic mapping? I feel like this is a really abstract concept for a lot of people, but it's actually not that bad. So most simply, it really just means placing something on a chromosome in the correct location. So I find it's a little bit easier to conceptualize this if you think about this with respect to a physical map like we are used to seeing. So here's a map, Hyrule, from Breath of the Wild. Okay, and so using this map, we can think about what does something mean? What are we going to place on the chromosome? So something could be a lot of different things. These are just basically landmarks. So this could be something like STR CODIS markers, which you know are used to identify individuals. These are what are going to be used in the FBI. So for us, things like CODIS markers, they're not really the thing that we're so interested in. They're just something to get us to the result that we need. So this could be, for example, driving through all of West Texas. You don't necessarily need to stop in West Texas, but you know it's going to get you to your destination. So on this map, this could be something like Rukuku Prairie, okay, which is hanging out over here. Obviously not a whole lot going on here. You may have to pass through it to get to your destination, and you may use it to place yourself relative to what's going on around it, but it's not really the thing you're trying to discover. So other things that we can think about in terms of being kind of more of a destination, we could think about genes that are important for a particular phenotype. So this could mean anything that we're interested in, pretty much at all. So you could think, for example, we see maybe some corn that's going to be very drought tolerant relative to another type of corn. And we want to know, hey, man, what's going on with you, corn? Why are you going to be drought tolerant? We want to know what are the genes responsible. So not just where am I, just any old place, but we want to figure out what the heck is going on here. So in terms of a destination, that might be more like Hyrule Castle. Okay, so that's kind of the ultimate destination you want to get to. You may pass through a few of these other places to get there, but this will be kind of really the big destination you're trying to get to. So we're going to be trying to actually place things on chromosomes exactly the way that we might place things on just a regular physical map. Okay. So thinking about that, we can start to review some of the tools that we've been learning about all semester and put together this idea of how we're going to place these, okay? So taking a step back, we can think about tools that we will need. So all semester long, this is genetics, we've been learning about Punnett squares, or as I like to call them, Punnett squares. So Punnett squares are not really here just to give you extra work. They're here to help us understand some things about the cross that we've done. Okay, so setting these up, we're going to always be putting gametes on one side, gametes across the top. And what these are really about, the essence of these, is to tell us, hey, what do I expect the offspring to look like? What are their phenotypes going to be? How many of each phenotype am I going to get? So this is really important just in the grand scheme of understanding how genetics works, but really setting up this expectation is going to help us understand what's going on when we deviate from it. So thinking about the way that Mendelian genetics works, this is obviously just one trait that we're looking at. We have dominant, whatever A is, and recessive, whatever A is. Okay, but we know there are more than one trait. There's more than one trait going on for any organism. And so we can start to relate this to principles of independent assortment. Okay, again, something that you should be really familiar with. So thinking about this, just kind of reviewing it so that we can attach it back to this Punnett square or Mendelian genetics, thinking about independent assortment, how it relates to different chromosomes. So we know here's the homologous chromosome pair, okay, chromosome 1, 2, and 3, one set from ostensibly mom, and here's one from dad, okay? And these are homologous chromosomes because they share general content. So they don't have to be exactly identical, but they should have the same genes on them with some variation. So there's a lot of sequence similarity here. Okay, so we're going to be learning a little bit more about that in a minute, okay, in terms of those physical maps we're trying to build. So independent assortment, of course, refers to the relationship between this chromosome and any of these others. The idea being that just because the red one goes to the left side, okay, it doesn't affect where the second and third one, how those are going to go at all. You can see all the different options here. Here, red's always going to the left on the top one, but blue for 2 and 3. You can have, again, 2 and 3 independent of each other. And so the idea being that when you have two traits, what we can expect from traditional Mendelian genetics or Punnett square genetics is that their ratios, the phenotypic ratios, what you get for each trait separately, they're going to be unrelated to each other. So the ratios you get for segregation for genes on these two chromosomes are going to be independent of what's going on these other chromosomes. Okay, so again, setting up that expectation. So we have talked about a lot of stuff going on with these homologous chromosomes. Okay, so one of the things that we can start to think about is, again, there's not just one gene per chromosome. So what is going to happen when these genes are actually on the same chromosome? So checking it out, here's a solid set of chromosomes here. You can see that on the blue one, we've got dominant A. Okay, we're going to have dominant B, all right? And on our pink chromosome, we're going to have recessive, whatever A is, and recessive B. Okay, so that's cool and all. And what we're going to be doing here is thinking about what that means. Okay, so I think that probably just from principles of physics, it's pretty clear that as long as these are attached on the same chromosome, wherever the top half of this goes, you're going to expect the bottom to go with it. Okay, so we're going to be thinking about these sort of units, genes all in the same chromosome. So in terms of placing these on the physical maps, we're going to be worried about mapping on one chromosome at a time, so constructing these maps. So to relate this back to our high-rule map example, we're concerned about, again, each chromosome by itself. So if you think about chromosome 1, this is going to be our map of high-rule. Again, we're going to have our cuckoo prairie somewhere over here, high-rule castle here. They're on the same map, okay? So we've got everything kind of in relation to each other. However, what you won't see is Yoshi's house from Super Mario World hanging out somewhere over here in Hyrule. Okay, this is a separate chromosome. So when we're dealing with mapping, we really, for most basic points, only care what's going to be going on one chromosome at a time. So we'll get to full genome stuff a little later, but for now, focusing on one at a time. Okay, so headed back over to our chromosomes. Okay. When we're thinking about, again, the solid case where dominant A is sort of obligated through physics of being attached to go wherever dominant B is going, recessive A kind of obligated to go with recessive B. The idea here is really getting around why are we ever going to have a case where dominant A and dominant B won't go together, and recessive A and recessive B also may not go together. And so the deal here, which I really hope that you're thinking after 2/3 of a regular semester in genetics, is recombination or crossing over. So again, you can see these two inner chromatids, sister chromatids, the two inner ones from the homologous pair. These are going to be forming a synaptonemal complex all along. Okay, it's a lot more solid than this looks, but this is clear, better for clarity. So you can then see that the result of that is that we're going to end up actually trading some genetic material between the two. And that's where we, again, get these cases where dominant A is going to go with recessive B. Recessive A can hang along with our dominant B. Okay, these outer sister chromatids didn't participate in this particular case. We'll see some examples where they might in the future. Okay, so thinking about that, how does that actually relate to a map? Okay, thinking about how a physical map can be created by having this understanding. So when we think about this whole crossing over kind of idea, this whole recombination idea happening around here, we can go back and think about, again, the physical space of these chromosomes. So we have, in this example, whatever units you want this to be, we have this much space to have chances of recombination to happen. Okay, so let's hang on to that and compare it. What if whatever gene B is actually is hanging around up here instead of where its original position was? That would only really give us this much space to play with. Okay, so something that I hope is fairly straightforward is that the bigger space you have in general, the larger chance you have for recombination to occur. Here, you only have this bit of space. You would expect this to be less frequent. And so what we're going to ultimately be looking at in later videos is how to use that math to our advantage, how to actually figure out what's the frequency and translate that into sort of a distance. Okay, so that's what we'll be working up towards. Okay, but what we're starting with, and when you think about some of those traditional sort of experiments that people were looking at, really what we have to rely on is not these beautiful pictures of chromosomes where it is very obvious, okay, here's this gene, they're here on the same chromosome. Instead, what we're going to be relying on most of the time to start is the phenotype, so the actual percentages, the ratios of the offspring that we see. What are these phenotypic ratios? And that's why it's so important for us to understand the Mendelian expectations for this so that we can spot this when we see it. Okay, so we'll have to rely on the phenotype. We will have to look at phenotypes and figure out whether recombination has occurred. Okay, so to illustrate this point, we can start off with an experiment by Thomas Hunt Morgan. Okay, so there's a lot going on here, all right, and a little bit of spoilers, which we'll see in a second. Okay, so I think in these types of examples, since there's so much going on, best thing to do is kind of focus on this from the beginning. So this is the two parents, the original parents of this cross. Okay, a little bit of stuff to get out of the way in terms of kind of how these will be noted here, the annotation of these genes. This is a little bit different than we may see in our textbook sometimes. So here what we're going to see, again, is recessive traits that are going to be noted just lowercase letters. Okay, so here we're going to have a recessive trait that's going to be for white eyes and little tiny miniature wings. Okay, and this is a lady fly, so we'll see that she has two X chromosomes noted by these two bars. Okay, so here we're going to have a male fly. Okay, he has one X chromosome, and you can see the wild type traits, red eyes, noted here with a plus, and regular standard issue wings, again noted with a plus for wild type. This little guy is our Y chromosome. Okay, so this is looking at sex-linked trait, but the sex-linked part of this, the fact that it's on the Y chromosome, is not really the important part for right now. Okay, it'll limit what the Y chromosome can participate in, but really what this experiment will demonstrate is evidence for this idea of recombination and linkage of genes by looking at the phenotypes that we see in the offspring. Okay, so of course what we're going to do is make these guys hang out in the same glass jar until they ultimately mate, and this produces our F1 generation. Okay, so as you could expect, okay, we're thinking about the gametes that can be formed from our original parent. Okay, female fly here, all we got is recessive. That's pretty much it. Okay, from our male fly, if this is a female, we got dominant copies of these alleles from the father. Okay, so this is sort of heterozygous genotype that we're sitting on right here. Okay, for our male fly, all our female parent can pass on is this recessive set of both of these alleles for both of these traits. It's going to be from the female. Okay, and then what we're going to see is, again, we have to get a Y chromosome from the father in order to be a male offspring. So this is what we end up with. Okay, so we're going to be looking at this a little bit more depth to see what's going to happen here. Okay, so now it's going to be time to pause that video. So take this time, actually pause the video, please, and start to work out what offspring ratios you'd expect from crossing the F1. Okay, don't try to do anything fancy just yet. Think about what offspring ratios you'd expect. Not even really thinking about sex-linked, any of that. Okay, welcome back. Hopefully you actually pause the video, work this out for yourself a little bit. Okay, so what we can see going on here, taking our gametes, this is going to be a little different than the standard issue Punnett square that we're used to doing. Usually when we have two traits, we're used to keeping them completely separate, independent assortment for the win. But here, basically, we're not taking into account recombination, not taking into account anything weird yet. We have to move these as a unit. So that's why this X chromosome here is actually kind of helpful. So when we think about filling in our female gametes, okay, the ones that came from mom and the ones that came from dad across the top, here's what we can think, just moving these whole sets as a unit. Okay, so we got gamete 1 here. We're going to be able to pass on dominant copies of an allele for eye color and our wing morphology. Okay, our second gamete, here we go. That's going to be recessive for both traits. For male fly, all we got is an X chromosome that's got recessive alleles for both traits and a solid Y chromosome just hanging out, not really doing much yet. Okay, so thinking about how that's going to play out, we can start to get our gametes kind of mated up here, beautiful union, sperm and egg, to find out what our phenotypic ratios would be. So gamete from mom, gamete from dad, okay, carrying this on. All right, so we can see here, homozygous recessive. Okay, here, this is, again, we're going to get a Y chromosome, but also dominant copies of both alleles. And here we're looking at recessive copies of both alleles with that Y chromosome. Again, phenotype is what we have to rely on. So you saw the spoiler picture, there's going to be some phenotypic information hanging out at the bottom of this cross, okay? So thinking about how that's going to go down. All right, so phenotypically, we can translate this to red eye with regular wing, so wild type for both traits. Red eye with regular wing, male, okay? Wild type for both traits. Here, we'll be looking at white eye with a miniature wing, female fly. And here, white eye, miniature wing, male fly. So if you think about it, how is this actually going to play out, okay? We can see that this is kind of, if you lump red eye and regular wing, red eye, regular wing, kind of don't care about the actual sex of these flies. You can see that red eye, regular wing, white eye, many wing. Okay, this is kind of a 1-to-1 ratio. We can lump this. Here's a category, category 1, category 2, 1-to-1 ratio. Okay, but again, as we saw for just a brief moment when we started this cross, that's not going to be exactly how this played out. So, oh dang, we had 4 phenotypic categories if you counted the actual sex of the flies, only 2 if we're just looking at the eye and the wing trait. Okay, so hanging out over here on the left, let's just kind of break this down a little bit. What we're going to be, again, looking at here is going to be the idea that we're going to have, again, 1-to-1, looking just like the original parents. We're going to have our white eye, many wing, okay, and our wild type set. Here's our white eye, many wing, wild type set. Again, if we ignore the fact that some of these are going to be male, some are going to be female. Okay, and kind of lump these together. You can see that we're kind of sitting at a 1-to-1 ratio, okay, here. But we've got, again, all these extra guys. What the heck, okay? So before we get over here to the right, what's happening here, we've got to think about, again, what is the phenotype and how does it relate to the actual parent that we started with, okay? So again, remembering that the parent we started with was a wild-type male fly, okay? So we had wild-type wings, wild-type eyes, just straight up wild-type for everything. And we had a female fly that had white eyes and miniature wings at the start of all of this. This is the same phenotype that we see in this set of offspring and in our Punnett square. Okay, again, no crossing overs occurred. We match, for the two traits that we're interested in, we match the parents. So these are called parental phenotypes. Again, red eye, regular wings, just regular old wings. Okay, these are going to be like one of the parents. The little white eye, miniature wing, one over here, that would be like the other original parent. Again, these are the parental phenotypes. Okay, but we can look over here to the right and see that's not how this played out. All right, so let's investigate this right side of what's going on a little further. All right, so let's take, again, the gametes that we could produce, the actual genotypes that we're looking at over here. Okay, and we can see when we're looking over here that we're going to just have this labeled red so that we can tell it apart. There's nothing different from what's going on over here. I've just labeled it so we can keep track of what's going on. Okay, so when we start to look at what's hanging out over here, we know, because again, it's 2020, we understand what recombination is, that we're going to have a crossing over event happen between these two genes. Okay, so it's going to look something like that. All right, so we're going to end up with a wild type allele for eye color with a recessive allele for many wings. Okay, that is going to be different than the original parental phenotype. Okay, here we're going to be looking at recessive white eye allele paired now with, on the same X chromosome, wild type wing morphology. Okay, so you can see how that occurred and how we sort of mix and match these traits. Okay, this is called decoupling. We are decoupled from the original parent state. Okay, and so this is going to help us figure out the rest of what's going on here. Okay, and so hanging out from whatever's going on with our male fly, listen, there's not going to be recombination here because the Y chromosome is a different chromosome than the X chromosome. They're not going to mess around together. Okay, they're just hanging out, ready to see which one you get. But we can use these to kind of finish up this Punnett square. So you can see that we can actually take our new gametes, new potential gametes that are formed. Let's just add them on to the bottom of this Punnett square. Okay, so you can see our wild type eyes, many wings, white eyes, wild type wings, and again, keeping the same male gametes hanging out across the top. Phenotypically, you can see that's going to give us the rest of these categories. We can have our wild type red eyes with our many wings, both female and male. Here we go, wild type eyes, miniature wings, and white eye, regular wings, okay, or wild type wings, okay, both male and female, okay. So this gives us those 8, if you're, again, kind of separating out male and female, are 8 phenotypic classes that you actually see, okay, and we can break these down into the sort of one-to-one here. These flies are said to have recombinant phenotypes, or honestly, just a shortcut, we call these flies recombinant, because the phenotype with respect to these two genes that we're interested in is going to be decoupled from the parent. So it doesn't matter what you start with, doesn't matter what the original state was. What is important is in the F2 offspring, whether the traits are going to be coupled or not. So these do not match what happened in the original parent, okay? So that helps us kind of fill out the rest of that Punnett square, okay? So this will become important. We're going to look at another example that's going to actually walk us through how to detect linkage with this, okay? And ultimately, again, not in this video, but in some of the next videos, we're going to be working towards placing these genes relative distance apart. So let's go ahead and work on that next example. Okay, so here we can look at another experiment detecting linkage through test crosses. Okay, so we're still dealing with flies because geneticists love flies because we know that we can do whatever we want and nobody's going to really care. Okay, they're really, you know, they really do produce a lot of offspring, so easy to work with. But here we're looking, instead of eye color, we're going to be looking at body color, where the wild type dominant trait is going to be a more gray color. Okay, and yes, this cross is in your book, but I just found these rando images from, I don't know, somewhere on the Google, so they won't exactly match the pictures of what's in your book, but they will match the cross that we'll see in a second. Okay, so our standard wild type dominant is going to be a gray body. Okay, whereas a recessive trait here, this recessive allele for this trait is going to give us a black body. Okay, we're then going to see that we have two sets of wing morphology here. We're going to have wild type wings, again noted with the plus, and vestigial wings, which is going to be the recessive trait. Okay, so we're always going to set this up looking at our actual F1. Okay, so obviously this guy, when the plink goes out, is going to end up being heterozygous for both traits. Okay, so what we're going to end up doing is then performing a test cross with this F1. Hopefully you remember the test cross is going to involve an individual that we know to be homozygous recessive for all these traits. Okay, so we're going to set up this test cross, and now would be another great time to pause that video. What offspring ratios would you expect from doing a test cross with this F1? Welcome back. So you can actually see how this is going to end up. Hopefully you set up a little Punnett square, something like this. Again, having a little Punnett square down here. Okay, so this is our test cross. This is the same one that we were looking at before. Okay, we had the F1, which we know to be heterozygous for both of these traits. It's going to show the dominant phenotype, wild type phenotype for both. Our test cross, we're going to have a black body and vestigial wings. Okay, we know this guy to be homozygous recessive. So setting up these gametes, it's homozygous recessive individual. We've abbreviated this because, listen, all you can get is a recessive allele for either of these. So you can expand this out 3 more times and get a whole 16 square Punnett square. I'm gonna abbreviate it, okay? So then all we have to worry about is what's going on with this other parent. So we can have our dominant gray body color paired with dominant wing morphology, the wild type wing morphology. We can have that dominant body color with the vestigial wings. We can end up taking the recessive allele for body color, which gives us a black phenotype, and we can pair that with wild type wings, okay, allele for that, and we can take that same recessive allele for body color and pair that with the recessive vestigial wings. So these are the potential gametes we can work this out with. Okay, so that would give us, in terms of offspring, there we go. Okay, so we can see how this is going to actually go out. So 2 things to take from this. One, obviously we're getting expected ratios. Always going to be part of the point of doing a Punnett square. The second thing that we have to think about is, of these categories, which ones are going to match the parents and which ones are going to match their own selves. Okay, they're not going to actually match anything. So take a second, think about that. Okay, so we can kind of think about what exactly we're looking at here, which one's going to be the parent, which one's going to be our recombinant set of flies. Okay, so our parentals, we should have our black body with normal wild type wings, and we should have a gray fly that's going to have vestigial wings. Okay, so that's what we should expect for the parents. Our recombinants are going to be decoupled from that. So we can look at that in our next slide. Okay, so again, this table's from your book. I've kind of outlined with more detail the cross that they set up. Okay, so we can see from this, we have our parental category, our recombinant category. Okay, we've done a lot of chi-square tests in this class. Okay, hopefully you know how to do them, or maybe your test didn't work out so well on Tuesday. Okay, however, we can kind of see how they've collapsed this into parental and recombinant. Okay, so again, our parental is going to be gray vestigial and black normal. Okay, so they've kind of collapsed these two categories together. The recombinant would show decoupled phenotypes. This can be our gray, normal, and black vestigial. Kind of added those together. You can see this is how they add up down here. This is our observed number. Okay, so you don't have to worry about this. There's nothing you can do about the observed number of flies. That's just as nature intended. Okay, it's just the number of flies that there are. Okay, but what we care about is the expected ratios. So if we think about that sort of abridged Punnett square that we just solved, okay, we saw that we're going to have a 1 to 1 to 1 to 1 expectation for these, or if we are going to lump together parentals and recombinants, a 1 to 1 expectation. So you can see that we've got here our expectation from our total 3,236 flies. Okay, so I don't want to be the one that counts those. All right, we're going to have a 1 to 1 ratio that we expect. If you are uncomfortable with collapsing the phenotypes into parent and recombinant, you can absolutely do it the longhand way by including each phenotypic class. Okay, it'll work out the same way in terms of your chi-square value, but you're going to have to remember that your degrees of freedom is going to be a little bit different. Okay, so here what we're going to look at, again, our observed minus expected. Okay, we're going to square that. Okay, and then we're going to divide by the expected. Again, this is just a chi-square test. This is the chi-square value. Okay, so hopefully this makes you feel a little bit better about the value that you may have gotten on the last exam. All right, and hopefully you can kind of eyeball this. Obviously, we'd want to compare this to our chart to be able to tell what this actually means. But this is such a huge number that if you've even looked at that table recently, you pretty much know that with a number this large, we're going to be able to reject the null hypothesis, meaning these are going to be linked or something's going on here. Okay, so again, kind of adding this back in, this idea of recombination, thinking about recombinant phenotypes, adding this back into our chi-square kind of analysis that we've been looking at. Okay, so kind of reviewing some of these tools. Okay, so we've just got one more experiment to get through. This one's a little bit shorter because I just have a few points I want to illustrate here. Okay, and this is the Curt experiment. Okay, and this one I think there's a lot of stuff going on with this experiment. This is an extremely kind of weird experiment, but it's kind of beautiful in that what we're going to be actually doing is linking physical properties of the X chromosome with the actual recombination event. Okay, and this is important because again, for us, this is 2020. Okay, we understand that people can look at chromosomes. We understand what recombination is. We don't need people to spell it all out for us yet again. But thinking about this, this is one of the first times that you could actually visualize that sort of mix and match thing in real chromosomes, okay? And so this is going to deal with two traits that, again, are X-linked. Eye color, which we love to deal with, but it's going to be a little bit different this time. and eye morphology. We're going to be looking at the bar eye, which we've actually talked about before, okay? So eye color, eye shape, okay? So we're going to, again, just like usual, we're going to cross the two parents with differing phenotypes. You can see why we do a lot of this work in things like flies, where nobody's going to get all upset if we start breeding them and then breeding them to their brothers and then breeding them yet again. Nobody's going to care, but we're going to cross these two parents. And then we're going to compare the physical chromosome with the actual phenotype, okay? So the important part of this to understand is that we're going to be dealing with some weird X chromosomes, okay? So these are not going to be standard issue X chromosomes. There's going to be some stuff going on with them, okay? So this is going to be kind of an expanded view of a normal X chromosome. Here, we've got a recessive allele for the eye color, thinking about kind of this carnation eye color. Here, we've got a dominant allele, okay, our wild type allele for eye shape. That's going to give us these nice kind of almondy shaped eyes as versus kind of this bar eye phenotype, okay? So that is okay for now. This is just a normal X chromosome. I actually just pulled this from dad's gamete over here, okay? But let's check out what's going on with Lady Fly, okay? Notice these weirdo X chromosomes that this particular fly has. We've got kind of an abbreviated version of an X chromosome going on here. There's a little detached piece that is going to go somewhere away, okay? It's going to be detached. It's going to be gone. We're going to lose it. It's gone, okay? Then we can put this guy over here, save it for just a second. The other chromosome, at first glance, is, again, kind of a normal chromosome. We have some differences in alleles, but that's not the weird thing. Okay, similar length. We don't have this weird detached piece like we saw over here. But instead, we're going to have a giant chunk of the Y chromosome from somewhere that at one point or another got attached to it. So imagine you can actually look in the microscope and be like, okay, little baby X chromosome and mega X chromosome with a little bit of Y on it. Okay, as in Y, are we counting all these flies here? Okay, so we have these weird things, and the fact that they look different is going to be where we actually see this happen. So we can focus on the parental. That's what we've got here to start, okay? And then we can think about these gametes we get through recombination between the gene for this eye color and our bar eye phenotype, okay? The gene for that bar eye phenotype. So we're just going to do a little mix and match here, okay? And so imagine you're looking through this microscope. You're like, okay, some of these gametes are what I expect based on what she's got to work with up here. But all of a sudden, here is a normal sized X chromosome. Looks normal. But in return, we've got a little baby short piece that's got that little Y chunk attached, all right? That's different. You can see we just did a mix and match, okay? So what's going to be important here is, again, comparing this to the phenotype. So when you look at chromosomes in the microscope, they don't have all these annotated, oh, here's this gene, check it out. They don't label themselves as being wild type or dominant or any of that mess. So what we're going to be seeing are the actual gametes that come out of this. And we're going to be comparing that ultimately, again, the gametes that are possible here, okay, with our parental category and our recombinant chromosome category, again, thinking about the chromosomes. If we had to do this by phenotype, we got to look at that, okay? And this one's very complicated. First of all, again, it's sex links. We're dealing with sex-linked traits. Second of all, what we're going to see is that our bar phenotype is going to have kind of an incomplete dominant sort of a situation. So you can see like little baby bar eye, kind of like jelly bean eye, and then standard almond eye. So we've got some of that going on, okay? And then our eye color, that one's a little better. We've either got carnation or your red. That's pretty much what we've got to work with, okay? We've also got this idea of male and female. Okay, so it's important not to focus too much on, again, the sex-linked part of this, but again, we can kind of focus on first kind of what's going on with these female. They're going to get a normal X that the male fly had up here to begin with, okay? But we've got this kind of set of parental weird X chromosomes and then our recombinant ones, okay? So we're going to, again, be dealing with this sort of split. We've mixed and matched between these genes, okay? So our parental ones, okay, we started out, again, with kind of our little jelly bean shaped eye, okay? And we're going to get, again, the recessive allele, so that carnation color, same thing we get up here, we get our carnation eye. If we get the other one, we're going to have wild type for both alleles, okay? So we end up, we're going to have our regular, normal, all around standard issue eye. Okay, we can see then how these are going to be in the recombinant, kind of linking, okay, hang on, we've got a normal sized eye, which would imply that we've now got 2 wild type copies of this gene, but all of a sudden, we're not coupling it to the red color anymore, okay? So that's where this is going to be decoupled. Again, we can see how this is going to end up. All right, our recombinant chromosome and attaching it to the phenotype, okay? So this experiment, I really like to show this, not because I think it's important for you to be able to anticipate what the eye morphology of every single thing that could possibly go wrong with a fly eye would be, but I think that it's very cool because it actually illustrates how we can physically watch this recombination event sort of unfold, because we have these sort of markers, this little tag from the Y chromosome, and because we have this like shortened piece, okay, we can kind of tell physically what's going on. Okay, so just to go ahead and conclude this video, what we've learned about today, mapping, putting genes together, we've got to think about this in terms of linkage groups, or what's in that same chromosome, what's going to be on that same physical map. Okay, are you in Hyrule or are you in Super Mario World? Okay, you just got to know, and that's going to be informed by this idea of linkage. And we're going to ultimately be taking advantage of the varying frequency of recombination between these linked genes to tell approximately how far apart they're actually going to be, okay? So it's going to be kind of like thinking, okay, it takes me 5 hours to drive from Shreveport to New Orleans, all right? But if there's traffic in Baton Rouge, it's going to be longer, okay? So the distance is going to be the same, but kind of what happens between, we'll see kind of relating that to recombination, okay? We can also, from the Curt experiment, we've been able to compare physical and genetic properties of these chromosomes, actually see how they're going to undergo crossing over using these kind of weird, visible abnormalities in these X chromosomes, where this crossing over actually is going to take place. Okay, so next time in our next video, we're going to start learning to use this recombination to find an actual relative distance between these 2 genes. So we're going to start putting all these things together. We've kind of taken that journey where we're going to be thinking about, okay, what Mendelian ratios did I actually expect from these genes? Did they assort independently or were they linked? Okay, we've done those chi-square tests, taking time to demonstrate mathematically that that was the case. Okay, and we know that they're linked. Now we're going to use that information to actually start putting together that map. So that will be what we can expect for our next video. And just to end today, I will leave you with my biology joke of the week.